Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad AIZAT and my members AWFIYAH. Today, we will discuss about practical 1, basic concept of statistic and variation for our subject, which is BIO510 ecology. Objective for this practical one, firstly, to calculate and describe the following descriptive statistic, means, variance and standard deviation. Secondly, to understand the concept of correlation. Lastly, to determine the relations between the two variables.
There are 4 sets of data and analysis graph that we obtain in this practical. Number 1. Discuss the difference or similarity of the mean of length between different set of leaf. Do the same for the mean of width. For question number 2, what is meant by variance? The leaf have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance what is meant by standard deviation. And the last question is, why is it necessary to have different set of measurement for the measurement of leaf, width, and length? Width and length.
Based on the result shown, set 2 and set 3 have the highest value for the length compared to set 1 and set 4. Furthermore, the leaves width for set 2 and set 4 have almost the same measurement, while set 1 have the highest value of width. In this practical, the size of leaves are different. This is due to various factors, which is amount of sunlight, soil nutrient, age of the plant, and water availability. These factors interact in complex ways, and the size of leaves is often a reflection of how well a plant adapts. For conclusion, we can say that the objective of this laboratory practical is achieved. This is due to we have learned the basic concept of statistic and variation. We also learn how to calculate the mean, variance, and standard deviation. Lastly, we understand the concept of correlation and relationship between the two variables.
There are 4 objectives in this practical. Here are the materials we use while completing the practical. In our practical, different types of forests in UiTM Jingke was the area under study. Here is the result for Group 1, Group 2, Group 3, and finally Group 4. Moving on to the next one, which is post-lab question. The first question is, define the term quadrat in practice. Quadrat is a square or rectangular plot of land used to mark off at random physical area to isolate a sample and determine the percentage of vegetation and animals occurring within the marked area. To calculate plant frequency or frequency index, you simply need to note down whether the target species is present or absent within each quadrat sampled. The number of quadrats in which the species was present should be divided by the total number of samples taken and then multiplied by 100 to get the frequency as a percentage. For example, in a survey where 10 samples were taken, dandelions were found in 6 of these. This would give a frequency index of 6% multiplied by 100 equal to 16%.
Second, what are the 2 basic requirements of all the quadrat technique? The 2 basic requirements all of this technique are that the area or volume counting is known so that the density is determined directly. Secondly, the organisms are relatively immobile during the counting period, so none are missed.
The third question is, what would be the advantages and disadvantages of using larger or smaller quadrat to survey the sedentary organism? The advantages of using a larger or smaller quadrat to survey the sedentary organism are: it is less expensive than other methods because it requires cheap equipment. Larger quadrat is a great representation of the population as it covers more area and the accuracy of estimation of overall density increase. However, it is also time consuming to place and sample. Meanwhile, the smaller quadrat is more time efficient and fine-scale patterns of sedentary organism distribution, like microhabitat preference, can be captured better. Even so, smaller quadrat might result in underestimation of population size as there are fewer individuals to be counted. In short, the decision regarding using larger or smaller quadrats is based on the specific study's aims, the feature of the sedentary organisms under research, and practical factors like survey resources and time availability. Every quadrant size has advantages and disadvantages, and researchers usually choose the size that best fits their objectives and limits.
The experiment used quadrat sampling to identify and measure plant distribution in a specific area. A 5 meter times 5 meter quadrat was created using string and stick and randomly put in various areas. All plant species were observed and recorded, and their distribution within the area was estimated. Based on the result, it is found that species A dominating all areas that being observed. This is because species A can grow in many types of soil, even acidic one. It spread naturally by its seed being carried in animal dung or sticking to the fur of grazing animal. This plant does well in tropical and warm subtropical climate, which make this area a perfect place for it to grow. Its ability to adapt to different soil and reproduce easily help it thrive in this environment.
In conclusion, the quadrat sampling experiment effectively introduced the concept of quadrats, enhanced observational skills, recorded plant distribution differences, and identified factors affecting plant distribution and abundance. Accurate and reliable results were achieved, showing that species A dominated the observed areas due to its adaptability and natural reproductive methods. The precautions taken ensure that accuracy of these findings, demonstrating the method's effectiveness.
Now, let's move on to our chosen species, which is clownfish and anemone. So basically, for your information, clownfish is omnivorous. They primarily feed on algae and plankton, but also consume small invertebrates such as zooplankton and various types of small larvae. Algae give them essential nutrients and contributes to their overall health, while plankton is a crucial part of the diet, especially for juvenile clownfish. Larvae are rich source of nutrients, and they also consume detritus. When they consume these things, it helps in recycling nutrients within the ecosystem. And lastly, they also consume leftover food from anemones from the symbiotic relationship. Next, let's move on to defense mechanisms of clownfish. There is 6 defense mechanisms, which is symbiotic relationship with sea anemones, mucus coating, camouflage, quick reflexes and agile swimming, social structure, and territorial behavior. First defense mechanism for clownfish is symbiotic relationship. It happens when sea anemones provide protection for clownfish. Sea anemones have poison on its tentacle, so it can protect clownfish from the predators. Next is mucus coating. Since they live in poisonous sea anemones, clownfish have mucus that coating them to protect them from sting of anemone's tentacle, so it allows them to live safely among sea anemones. Not only that, it also can camouflage. Vibrant colors of clownfish can help them blend in with the similar colorful sea anemones. Clownfish have quick reflexes and are agile swimmers that can dart quickly into the safety of anemone's tentacle when they are threatened. The social structure of clownfish is living in small groups with strict social hierarchy. This organization helps them respond quickly to threats and provides a structure for communal defense. Lastly, the territorial behavior, which is clownfish are territorial and will aggressively defend their anemone's home from intruders and potential predators.
Alright, just now Syakirah already present about the clownfish, but right now I am Is and will present about sea anemone which have the symbiotic relationship with clownfish. But before we talk about that, let's talk about the first mechanism of sea anemone first. Sea anemone have a sting and tentacles that called nematocysts, which are able to inject toxins into predators and prey. This make it difficult for predators to approach the sea anemones without getting stung. Next, sea anemone can secrete mucus that contain toxins or other deterrent chemicals in order to keep predators away from eating them. And lastly, when the sea anemones getting threatened, they can retract their tentacles and contract their bodies to become less visible and less risk of injury. Some anemones can blend in with their surroundings, so it will make it difficult for predators to spot them. Alright, let's talk about symbiotic relationship of clownfish and sea anemones. The relationship between clownfish and sea anemones are mutualism, where both species gain advantages from the interaction, which can be either food, protection, or other resources. So how both clownfish and sea anemones can give benefits to each others? So, sea anemones can protect clownfish using the nematocysts, which can kill or paralyze other fish. Since clownfish have a special mucus on their skins to protect them against stings, this has provided them with the safe habitats where they can hide from predators. Sea anemones can get benefit from clownfish ability, which attracting other fish with their bright color. As they approach, they get stung and eaten by the anemone. Clownfish may also feed sea anemone by scavenge around them and may feed it with small particles food. Clownfish also clean the sea anemones by removing debris and also parasites, which keep the anemones healthy and infection-free. Okay, lastly, clownfish movement through sea anemones tentacles increasing water circulation around the anemones, which can help with respiration and waste product elimination. So I think that's all about clownfish and sea anemones. Bye!